This tutorial will introduce you to the Dataset Explorer, which allows you to search and filter datasets to analyze. To start, click on Dataset Explorer on the top left of the menu bar. From there, you can enter a search criteria in the search bar, for example, preeclampsia. Your results will be sorted by relevance. For more details about search, you can hover your mouse over the question mark icon. It describes how you can search for an exact match using quotation marks or combine words in a search using the AND operator. You also can select a term from the autocomplete dropdown in the search bar. You can narrow the scope of the search results using the filters in the left-hand navigation pane. Select Expression Profiling by Array from Experiment Type and Human from the Species filter. You can click View Details to find additional information about each dataset, and can click the green Add to Workspace button to save datasets to your workspace. Note that at any time, you can use your browser's back button to return to the previous page. Once you have found an interesting dataset, click the title to go to its Dataset Overview page. The Dataset Overview page displays additional metadata, supplementary files, and samples, along with the PCA widget to visualize the structure of the dataset. The sample selection is the entry point to the analytics. You will select control and experimental groupings to analyze. If you click Show Attributes, you can select or unselect which columns are displayed. If you click Filter Rows, you can search and filter within the columns. You can also hover over cells or expand the columns to view all information. In this case, we will select samples with title Chorionic villi EOPET for early onset preeclampsia, and click Define Groups to add them to the experimental group. Then we select samples with title Chorionic villi Control and add those to the control group. Once you are satisfied with your selections, click Run Analytics. The tool will begin analyzing your selections. Note that this may take up to several minutes, and the progress is indicated near the top of the screen. At any point, you can click the Metadata tab to view and change your sample selections and rerun the analytics. The first step computes differential expression and displays an output table containing relevant statistics. Genes are listed in order of increasing p-value. The table can be searched by clicking Filter Rows. You also can click Generate File to download this information. In the next step, the Clustergrammer tool generates a hierarchically clustered heat map of samples versus genes. The heat map is interactive. You can change the order of the columns and rows alphabetically, by cluster, by sum, and by variance. You can search within rows to find specific genes of interest. You can perform dimensionality reduction by filtering rows using the top sum and top variance sliders. In the top left corner, there are additional tools for interacting with the clustergram. You can use the crop tool to zoom in on certain areas of interest, use the download icon to download the matrix of data in the visualization, or use the snapshot icon to take a SVG or PNG of your visualization. You also can click on the gene icon to perform enrichment analysis to find biological information. For example, if you select KEGG 2016 as your enrichment library, you see the top 10 enriched pathways. In the next step, correlational and covariance PCA analyses can be used to identify outlier samples. The system computes PCA and displays an output table with cumulative proportion, proportion of variance, and standard deviation, and two plots, a scree plot and a biplot. You can specify covariance matrix or correlation matrix. Gene-set enrichment analysis can identify up and down-regulated cellular phenotypes. The system performs a gene-set enrichment analysis against the gene ontology and displays a list of significant genes with IDs and definitions. you can adjust the P cutoff and specify P FDR or P and FDR. The next step displays an interactive network diagram of significantly expressed genes and their gene ontology terms. You can move nodes and click on them to investigate specific network connections. Pathway analysis can be used to identify and visualize significantly up and down-regulated pathways. The system displays a table of pathways with relevant statistics. If you click on any of the entries in the table, the system displays an image of each pathway retrieved from the Reactome database. You also can click Generate File to download this information. This concludes our analytics tutorial. Please contact PAT user support if you require any additional assistance.